All right. Can you see my video now as well?
Yes, I see your video too.
Perfect, thanks. Alrighty, so get started. I'm Amy Goldman, and James Stegen and I will be presenting today an introduction to WHONDRS. I'm an earth scientist at PNNL, and my pronouns are she, her. And I'm going to go over, there we go. I'm going to go over the background and motivation for WHONDRS and an introduction into the sampling campaign that produced the data that Tim mentioned that will be used for this course. And then James will take over and we'll go over some of the data from that campaign as well as some high-level objectives. So some of you might be familiar with WHONDRS. I want to make sure that we're all kind of starting out with the same basic knowledge of it and that everyone understands how they can engage with it and how it can be a resource for you. So if you see on the right there, WHONDRS stands for the Worldwide Hydrobiogeochemistry Observation Network for Dynamic River Systems. And we are a consortium of researchers studying river corridors all over the world. We collect physical, chemical, and biological data and make all of those data open access. And we really model ourselves to be a resource for the community. So the broad aim of WHONDRS is to accelerate transferability of knowledge, data, and models via open coordinated science. And one of the ways that we do this is by creating studies and then shipping out free sampling kits to collaborators that allow us to collect samples all over the world. And so this map is from our summer 2019 sampling campaign. And because of our amazing network of collaborators that volunteered to collect samples, we were able to collect from 97 sites around the world, which ended up producing over 7,000 subsamples for different analyses, which is pretty amazing. So WHONDRS is able to work towards this goal because it's built on a foundation of icon-fair principles. And this might be a new acronym for you, so we'll kind of go through it step by step. But WHONDRS is integrated in that we couple chemical, physical, and biological processes across both spatial and temporal scales. We are coordinated in that we ensure we're using consistent methods and protocols to enable transferability of the model relevant data that we intentionally target. And one of the ways we do this is through the shipping of those free sampling kits that I mentioned. We also write super detailed protocols and we film video protocols and we make sure that collaborators have a lot of opportunities to engage with us to kind of make sure that everyone feels prepared to have things be as consistent as possible when they're doing any sampling. And then we are open across the whole research lifecycle. So early on, we share our study design with collaborators through webinars, telecons, and we try to get feedback and people's ideas on our study design. And from these interactions, we've changed our methods. We've also added different data types, and that's been at times because collaborators have volunteered to do different analyses that we hadn't planned. And that's, enriches our data sets, and it also adds data types that they're most interested in. So it's been a really valuable component of engagement for both us and our collaborators. And then, of course, we're open as well because our data are all openly accessible. We publish our data on ESS Dive, which is DOE's data repository for Earth System Science data. And I'll provide some more information about that in just a moment. But we don't hold back our data prior to publishing it. We're not trying to use our data before anyone else can get it. We really just hold it to do QA/QC, and then we publish it immediately after. This can still, of course, take some time, and so we also post preliminary data up on a Google Drive so that people can have access to it as quickly as possible. And then we also are open in that we are consistently structuring our data. We are working towards fair data practices so that our data are findable, accessible, interoperable, and reusable. And so we are then finally networked in that the community is completing the sampling as I described. So instead of us having to travel around the world, we are able to really get these global data sets because we have these volunteers from the community who are willing to collect samples. And this, benefits us in these global data sets, but it also allows our collaborators to learn more about their local systems and access resources that might be difficult for them to access. And that includes some of the data types that we'll be going through during this course. So these ICON FAIR principles and this approach are really a realization of DOE's vision for open watershed science by design. So this is just the cover of that report, and WHONDRS really embodies this vision and is a use case in this recent workshop report that they put out. So I encourage you to take a look at that if you're interested in kind of trying to make your own work go in this direction, or if you're interested in learning more about this approach. So of course, as I mentioned, a big part of this is openly publishing our data. So accessing our data on ESS Dive is super easy to do. You just have to search WHONDRS into the search bar. The website is up at the top there. And when you put WHONDRS in, spell it correctly, and then you'll get all of our data packages listed. You just have to click on one, and we've structured all of our data packages the same so that you just have to download a single zip file, and then you'll get our metadata and our data in that. We've put quite a lot of metadata into our data packages to really make sure that everything that someone could want is in there. But of course, if you have questions or want any additional information, we encourage you to reach out to us as well. And then all of our data are published under the Creative Commons Universal Public Domain dedication. And that basically just says that anyone can use these data for anything. So this is a really important thing for us. What WHONDRS is really supposed to be a community resource. And so we want you to be using our data for your research, your proposals, presentations, anything. And so we want your interest to also inform what we do, and we want your priorities to inform what we do. And so these three figures are just showing a map of our current locations that we've sampled, and then a picture of some of our team creating the kits that we send out to people, and then just a screen cap of the inside of one of our data packages showing some of those files. And so if you are interested in kind of seeing some more of this laid out, you can take a look at our website. It lists all of our studies as well as our published and preliminary data. And it also has some different science questions that you can look at to sort of think about how to explore these data. But really, WHONDRS only works if the community is engaged. So we'd love for you to get involved. As I said, we have our website. You can also send us an email, whondrs@pnnl.gov. You can connect with us on Twitter @WHONDRS. And we would really love for you to consider submitting an abstract to this AGU session. Abstracts are due on July 29th, and the meeting will be mostly virtual this year. So there's no real reason not to submit one if you're interested. If you're interested in collecting samples with WHONDRS, there are a number of ways to get involved with that as well. I've posted some pictures here. These are pictures that some of our collaborators tweeted from our last summer sampling campaign. And you can see they seem to be having quite a lot of fun. So highly recommend that. But you can collect surface water samples using our little mini kits, we call them. They take like 10 minutes to sample. They can be used basically anywhere at any time, as long as they're near a stream gauge or some other way of measuring stream flow. You can also get involved in a future large coordinated campaign, like our summer campaign that had the 97 sites. You can get involved in an upcoming wildfire associated campaign if your site is impacted by wildfire. or you can help us support small sampling campaigns for individual researchers' field sites that we help to facilitate. So if you're interested in doing any of that, please reach out to us. We'd love to supply you with additional information and answer your questions. So with that, we can kind of zoom back into this week. So the data from this week come from a 2018 WHONDRS campaign. And so the map on the left there is showing the 7 sites that Tim had mentioned. And this was our 48-hour diel cycling study. So we aimed to target sites with different drivers of subdaily hydrologic variation. And so we ended up with 7 sites for this. And this was really to investigate connections among hydrology, chemistry, and biology under these very dynamic conditions, so that you could ask questions, for example, like whether these short-term fluctuations might drive dynamics in the chemistry and microbiology. So this is a map of our 7 sites that we sampled at. And again, a big thank you to our collaborators who agreed to do this pretty grueling sampling with us. So on the upper left there, we have the Columbia River site. The Columbia River is a really dammed river, and so this site is heavily influenced by hydropeaking behavior. And then moving clockwise, we have East Fork Poplar Creek in Tennessee, as well as the Erpe River in Germany. Both of those have hydrologic variation influenced by wastewater effluent. And then we have the Jordan River in Israel, which is influenced by irrigation pumping, the Altamaha River in Georgia, which is tidally influenced, but is still a freshwater river. And then we have Watershed 1, which is in Oregon at H.J. Andrews Experimental Forest, and that is influenced by evapotranspiration. And then lastly, Nisqually River in the mountains in Washington, which is influenced by glacial freeze-thaw cycles. So at each of these sites, we sampled pore water and surface water every 3 hours as water level varied over a period of 48 hours. So quite a lot of sampling for our collaborators to do. I've posted these pictures here just so you could get an idea of what this sort of hydrologic variation looks like at one of our sites. So this is our Columbia River site. At some of our sites, we only had a couple millimeters or a couple centimeters of change over the 48 hours. This site had some of our largest variations, and these pictures are a little bit misleading because in reality, this whole site flooded and we had to move the camera. So this is just to kind of give you an idea. You can see the yellow and red circles are just circling some stakes that we had in the ground. So we have the yellow on the left and the red on the right. And then starting on the left, we have at hour 0, the yellow stake is at the water line and the red stake is underwater. And then we jump ahead 3 hours to our next sampling time point. The water level has now gone down and the yellow circle is now sort of high and dry and the red stake is at the water line. And then if we jump ahead several more time points, you can see that the water level has risen at both of those time points and the stakes are kind of further and further underwater. And you can see the water level actually creeping up the legs of this table that we had to put out. So all of our sites use the same installation and sampling design. This is, of course, part of that coordination and consistency that I mentioned before. We sent out sampling kits so that everyone would be using the same method to approach this study. And so on the pictures on the right, you can see the Jordan River site. Towards the left of the pictures, you can see some tubing set up. So everyone installed 4 lengths of tubing in their site. This was to collect surface water from 1 tube and pore water from 3 of the sampling tubes. And then we just pulled water from those tubes with syringes. And you can see the timestamps on these photos from Jordan River. We have 4 in the morning and then 10 in the morning. So again, this is pretty grueling sampling. We're lucky to have people volunteer for. And so the surface water tube, we just kind of anchored near the riverbed. And then for the pore water tubes, we attached those to these push point samplers that we installed into the riverbed. So they're just these kind of thin lengths of metal tubing. You can see them on the right, and they have a little point at the end and a screened interval near the bottom. And so we installed them down about 10 centimeters, and then we were able to just pull up with the syringe to access that pore water. And so the water samples from these lengths of tubing, we collected filtered water for geochemistry and FTICR-MS, which we'll be hearing more about later if you're unfamiliar with. And then we also preserved the filters in RNA later for metagenomics and metatranscriptomics. And so all of these analyses were supported by the PNNL SBR SFA that Tim mentioned earlier, EMSL, the Wrighton Lab, JGI, and KBase. And of course, we also publish our data on ESS Dive. So with that, I'll hand it over to James to go through some of the data from this campaign and then to go through some high-level objectives for the course.
Excellent. Thanks, Amy, for all that. And let's see, so I'm James Stegen. I am PNNL. My background is in ecology. I co-lead WHONDRS with Amy. And so I'm just, as Amy was saying, I'm going to give you a really brief little overview of some of the data, just kind of whetting your appetite for things that'll come throughout the week. So if we start here on the physical side of things, and by the way, this slide and the next two were prepared by Bob Danzak, one of our colleagues. You'll hear more from him in particular tomorrow. So as Amy said, we focused on sites that had a lot of dynamic hydrology across the 48-hour or even shorter timescales. And so you can see the 7 rivers on the right. That's basically the height of the river across 48 hours. You can see they're all quite dynamic. The level of dynamics changes across the rivers. The qualitative patterns of the dynamics are different across the rivers. As Amy was saying, they're all driven by different mechanisms. Evapotranspiration, glacial melt, hydropeaking, etc. And so that's a theme I would encourage you all to think about, is that there are commonalities across these rivers, but there's also differences in the physical status, the physical conditions, the chemistry, the biology. So in some ways, we are interested in thinking about what is transferable, what's consistent, but also what's different, what's context dependent. Both sides of the coin are important. And I encourage you to think about that throughout the week. So then, Amy, next slide, please. And I'll show you just a little bit of the chemistry, just to kind of orient you a little bit to the basics of these rivers, so to speak. So here we've got dissolved organic carbon concentration. On the left-hand side, you can see the 7 rivers there. It's quite variable across the rivers. The Erpe River being the highest, up near 10 milligrams per liter. The Nisqually, that's a glacial river, is down towards the detection limit of our instruments, down around 0.2, 0.3 milligrams per liter. And then, so in addition to that, we have a variety of other geochemical variables that are measured, things like sulfate, sodium, magnesium, nitrate, etc. And so you can take those multivariate data and collapse them onto an ordination, which is shown on the right-hand side. If you're not familiar with these kinds of plots, basically each circle is a sample from one of the systems, and the closer the two circles are, the more similar that they are in their multivariate geochemical profiles in this particular case. And so you can see lots of things by looking at this. You can see that, the different colors are different rivers. The open circles are the pore water that's coming from the subsurface. The closed circles are the surface water data. So you can see, like over there on the left, the Erpe River is quite variable through time. The surface water and pore water are relatively similar to each other. The circles kind of in that purple color. The red circles are from the East Fork Poplar Creek. Those, you can see that the surface waters and the pore waters are very divergent in their geochemistry, such that the pore waters actually look like water from the Jordan River, which is, of course, very far away from the East Fork Poplar Creek. And so you can also see, some rivers are highly variable in time based on the spread of the points. Other rivers are have very little variation through time in terms of their geochemistry. The points are very clustered. So that is just like a really brief overview of the geochemistry and variability. And Amy, let's go to the next slide. And here we can see, start to get into the metabolomics a little bit. And so here, Amy mentioned FTICR mass spectrometry. You'll learn a lot more about this tomorrow. I'm not going to go into the details today. And this is, again, just to show that there are some similarities as well as differences across the rivers. On the left-hand side, this is another kind of ordination. And here you can see each river is kind of clustering with itself to some degree, but also has overlap with other rivers as well. And here again, if you look at the red circles there on the left, that's the East Fork Poplar Creek. And again, we see divergence between surface water and pore water here in the metabolite profile, in the chemistry of the organic matter. Whereas the Erpe, the kind of the purple magenta color, is very much overlapping between surface water and pore water, but is divergent from the others. But further, you can go there, the plot on the right-hand side. This is where we can start to connect some variables together. In this case, connecting the metabolomics data from the FTICR with the geochemistry. And so now on the right-hand panel, each circle, that is a profile from the FTICR data. But the lines there, the arrows, are from the geochemistry. And you can see that there are connections between some of the geochemical variables. You can see there's only a few of them plotted now, because not all the variables connect over to the metabolite profiles. But you can see, like the Erpe River in the magenta. Oh, there's some delay in the slides showing up. Okay, I'll do my best. I'm not sure how much delay that is, but do my best. So you can see the Erpe River is really connected to higher concentrations of a variety of variables, whereas the other rivers are much lower, in particular fluoride, changes in pH. You can see them kind of spanning a range of different geochemical conditions. And so again, this is just really just kind of scratching the surface. We'll get into all the dirty details throughout the week here. Amy, let's go on to the next slide. And hopefully there's not too much of a delay. So the main, and so, okay, sorry, if we kind of step back, that's like kind of digging in a little bit. Amy was telling you about the campaign, some of the science motivations. I'm giving a little bit of a sense of the data. But if we step back and think about what are we doing with that information? What's the purpose of this week? Tim and Nancy were giving an introduction to that. And I'll give a little bit a different kind of angle on that's a little bit more into the science side of things with respect to thinking about the broad, big umbrella. What we're trying to do is to link processes, link chemistry, biology, physical processes together, and to do that across scales to enable deeper understanding, of course, but also enable enhanced prediction, in particular river corridors and their responses to disturbances that take a variety of flavors and forms across the globe. And so that's a very large umbrella, and I would encourage you all, you're going to be presented with material for learning skills, thinking about science from a particular perspective. But the great thing is that we have, 356 people on the line right now, and I really encourage you all to think in a diverse way, in a broad way, go beyond the science perspectives that are provided to you by the instructors, because the power of something like this is to collect and bring together diverse thoughts, diverse voices, diverse people, to really help advance the science in a much more robust and fast and interesting way and useful way than we could do with just a small group of instructors that already kind of work on this stuff. So hope you all engage in this from your own perspectives. But I'll just give you a few examples of some of the perspectives of the instructors in terms of the science objectives. So that has to do with like generating new understanding of what molecular data tell us about environmental function. Deciphering metabolic potential, how that varies, connecting it to metabolic networks and how those vary across rivers. Linking genome-based metabolic inferences to other kinds of omics like metabolomics, geochemistry, and emergent biogeochemical function. Exploring the consequences of thermodynamics of organic matter for microbiology and biogechem and as well as biogeochemistry, and that bidirectional influence of microbes and organic matter. And really enabling comparative analyses across diverse river corridor systems. Amy mentioned this about driving towards transferability of our understanding and our models. And then lastly, being able to develop skills to link data with models and encode our understanding in models to enable enhanced prediction across scales. So Amy, could we please go to the next slide? And I'm looking at the time. I think we'll be okay here. So Tim already showed this slide. The contact information for WHONDRS is up there on the right. There's been some discussion in the chat already. I'd really encourage people to reach out to us by email, follow us on Twitter, check out the website for information. You can check out some of the preliminary data that's not on ESS Dive yet there. So please reach out. And I wanted to end with this slide just as an overview. Hyun Saab made this slide, a few edits from other folks as well. But you can see really what we're trying to do here is quite diverse, quite complex, but is also quite manageable. Bringing together biology from metagenomics, linking that with metabolomics, and the work to be able to do this has been the tools and the concepts that have been developed, really expanding over the last couple of years to be able to do this and to even link those very detailed molecular data over to predictive reactor transport models that bring together physical, chemical, and biological processes in the real environmental context. And so I think with that, we're probably out of time. Becky, I'm not sure if we have time for a question, but I'll leave it with that. Thank you. So I'm not sure, Becky, if we have time for a question or if there is a question.
Looks like I think you've probably addressed the questions that showed up in the chat.
Okay.
There was some questions about how we reach out to folks and whether we're interested in public outreach, and James, you had addressed that, I think, in the response. But definitely, it is something we are interested in. We so far have worked primarily with actual, with researchers, with scientists, not with public persons per se, but that's something we're interested in exploring. So thank you, James and Amy, very much for that presentation. And it is time now for us to move into discussion of metagenomics to be led by our colleague Kelly Wrighton from Colorado State. forward to seeing the 28 students in there soon. Thanks.